Welcome to Learn Yourself. Today we are going to learn and understand about cell organelles. In my previous videos we have discussed about nucleus, cytoplasm and all the basic characteristics and features of the cell. In this video we are going to discuss about cell organelles, and going forward we are going to discuss all the different types of organelles. They are endoplasmic reticulum, mitochondria, Golgi apparatus, plastids and lysosomes. So cell organelles are present into a multicellular organisms or the organisms who all are having an advanced cell structure or eukaryotic cell structure. Because in this cell there are n number of chemical activities are happening, and all those activities are different activities and they are based on the functions which the cell is performing. So all these activities needs to be separated among the specific region. So this region has been provided through organelles. So large cell needs a lot of chemical activities to support their complicated structure and function. To keep these activities separate from each other, the cell use membrane bound little structure, and that is organelles within themselves. So one cell and small organelles will be present over there, and they all are membrane bound, because in each and every organelles different types of activities are going on. We need to separate these activities because their product will be different and their function will be different. So to do so, they all organelles are membrane bound. And that's why they all organelles are membrane bound, so that they will be separated from each other. Eukaryotic cell do possess them. So this membrane bound organelles are only present in eukaryotic cell or the cell which are well advanced or the cell of multicellular organism. Prokaryotic cells do not have membrane bound organelles and they have nucleoids. They do not have nuclear membrane region also. Their very primitive cytoplasm, which is less organized, will perform all the activities for the cell. So that is a basic difference between prokaryotic and eukaryotic cell. Now for organelles, endoplasmic reticulum, mitochondria, Golgi apparatus, plastids and lysosomes are some of the examples. Going forward, we are going to discuss all these organelles in detail. We will see their structure and we will understand their function. Now let us discuss about ER. ER is a short form of endoplasmic reticulum. What it is? It is a large network of membrane bound tubes and sheets. It looks like long tubules or round or oblong bags or vesicles. So generally these are tube-like and vesicle-like structures, and there are two types of ER. That is, two types of endoplasmic reticulums are available into the cell. First is rough endoplasmic reticulum and second is smooth endoplasmic reticulum. Now these two reticulums are based on their surface. That ER has a rough surface, then it is termed as RER, and ER has a smooth surface, is termed as smooth endoplasmic reticulum. And based on their structure, that rough and smooth, their function also will be varied. So now we are going to discuss about the difference between RER and SER and why their surface is rough and why their surface is smooth, and based on that their function will also vary. Now as per the structures, there are two types of ER. First is rough endoplasmic reticulum and second is smooth endoplasmic reticulum. Both are important in terms of their function. With rough endoplasmic reticulum, the ribosomes are attached, and with smooth endoplasmic reticulum, this process is associated, that is known as membrane biogenesis. So both endoplasmic reticulums are very important for the cell. So let us see first rough endoplasmic reticulum. That rough surface is because of the particles attached to that surface and they are ribosomes. So ribosomes are attached to this ER and that is because that is rough. So this rough surface is because of the ribosomes attached to that. Now what are the ribosomes and what is the function of ribosomes? So ribosomes are protein manufacturing site. They send this protein to various places in the cell using ER. So this rough endoplasmic reticulums are having a ribosomes attached to that surface. So that's why their surface looks rough. And these ribosomes are actually protein manufacturing site. So ribosomes are manufacturing protein and they are manufacturing this protein and they are sending to individual places in the cell where and all they require the protein. So they are manufacturing and they are sending this protein with the help of ER. So there they are taking the help of ER to distribute the protein where and all they required into the cell. So that is the function of rough endoplasmic reticulum. Now smooth surface is there here and that is termed as smooth endoplasmic reticulum. What is the function of this smooth endoplasmic reticulum? That it helps in manufacturing of fat molecules or lipid. So they are helping in fat molecules and into the lipid. Now lipid molecules are used in various cell functions and as well as they are used in building the cell membrane. We know that what is a cell membrane? Cell membrane are the semi-permeable membrane which allows the transfer or the movement of material inside and outside the cell. So into the manufacturing of cell membrane, lipid molecules are important and those lipid molecules are manufactured at smooth endoplasmic reticulum. So what is membrane biogenesis? That smooth endoplasmic reticulum helping in manufacturing of fat or lipid molecule. So that lipid molecules in turn can manufacture cell membrane. So that complete process is known as membrane biogenesis and that happens at smooth endoplasmic reticulum. It also helps in detoxifying many of the poisons and drugs. Some of the substances which are injurious or dangerous for the cell, so those substances will be detoxifying by smooth endoplasmic reticulum. So this were the basic difference between smooth endoplasmic reticulum and rough endoplasmic reticulum. Our another important organelle is Golgi apparatus. It was first described by Camillo Golgi. So Camillo Golgi was a scientist who first saw Golgi apparatus and he describes its function and made a remark that Golgi apparatus is an important organelle into cell. Now what is a Golgi apparatus? That it consists a system of membrane bound vesicles arranged approximately parallel to each other in stacks called cisterns. So they are parallelly attached to each other and they are membrane bound vesicles. Again this membrane bound vesicles which are known as cisterns again joined with endoplasmic reticulums. Those endoplasmic reticulums earlier we have seen that they are having tubules like structure. So these cisterns are again connected with the endoplasmic reticulum and they are making more complex web of this tubule structure and this membrane bound vesicles. So why they are attached with the ER? Because the materials manufactured at ER is packaged and dispatched to various targets inside and outside the cell through Golgi apparatus and that's why they need to connect with the ER. Earlier we have seen that ER are two types, that is smooth endoplasmic reticulum and rough endoplasmic reticulum. At rough endoplasmic reticulum, the protein molecules are manufactured at ribosome sites, whereas in the smooth endoplasmic reticulum, lipid or fat molecules are being manufactured which are helped in membrane biogenesis. So all these materials which are manufacturing at ER sites, they are being sent to the different sites of the cell with the help of Golgi apparatus. So that's why This cisterns needs to connect with the ER. So the functions of the Golgi apparatus is to materials manufactured at ER is packaged and dispatched to various targets inside and also outside the cell. Then second function is this material requires storage, modification and packaging of the product, and all these three things are also happening at Golgi apparatus. Then third function is sometimes complex sugars are also made from simple sugar in Golgi apparatus. They are converting some of the simple sugars into complex sugars, and that is also happening in Golgi apparatus. And the fourth function is it involves in the formation of lysosomes. In coming segment we are going to discuss about lysosomes, that what are the lysosomes and what is the function of the lysosome. But lysosome is also one of the important organelle into the cell, and Golgi apparatus is involved in the formation of lysosome. So these are the functions of the Golgi apparatus, and it has been discovered by the scientist named Camillo Golgi, and based on this scientist name we have named this organelles Golgi apparatus, and they are making a structures called cisterns. Our another important organelle, that is lysosome. This is also known as suicidal bag of the cell. Now let us discuss about lysosomes, that they are membrane bound sacs filled with digestive enzymes. So they are membrane bound. We know that in animal cell all the different types of organelles are membrane bound. Membrane bound means that organelles will be within the membrane. So whatever material they are producing or whatever activities they are doing, that will not be mixed up or they will not interfere in another organelle's function, or those material will not be mixed with another organelle's material. So that's why each and every organelle in animal cell are membrane bound and they have their own specific region. So these lysosomes are bags and they are filled with digestive enzymes. These digestive enzymes are made by rough endoplasmic reticulum. So the enzymes, the digestive enzymes which are present in lysosome, that is manufactured by rough endoplasmic reticulum. Now the functions of lysosomes are, they are waste disposal system of the cell. This helps to keep the cell clean by digesting any foreign materials entering into the cell, for example bacteria or food. Through the cell membrane, when any material or bacteria which are harmful for the cell, they are entering into the cell, so this digestive enzyme present into lysosomes will digest them and they will help cell to survive. So they are waste disposal system of the cell. Sometimes food particle also they will digest it if the food particles are not good for the cell. It breaks down complex substance into the simple substance because they have powerful digestive enzymes. During the disturbance in cell metabolism, so when cell is performing any activity, any function, during that time if some disturbance will happen and due to that the cell will get damaged, then lysosome may burst and enzyme digest their own cell. So lysosomes will burst out, enzymes will come out and it will digest its own cell because due to the disturbance cell has been damaged and now that cell is no more functionable. So that's why this lysosome will burst and its enzyme will come out and it will digest its own cell. So there will be no waste will remain after this cell will get digested. And that's why they are known as suicide bag of the cell, that it will eat up or it will digest its own cell. So that's why it is known as suicide bag of the cell. So in turn lysosome help cell to keep the cell clean because all the poisonous substance and waste substance will get digested. Along with that it will also digest some of the complex substance and it will convert it into simple substance. So in turn, lysosome is very important organelle of the cell as it will keep the cell clean. Our next important organelle is mitochondria. It is a powerhouse of cell because it has ATP. ATP is adenosine triphosphate, and this molecules generally cell is using when cell requires certain energy to perform a function. So it is a powerhouse of the cell. It has two membrane covering. So mitochondria has two membrane covering, that is outer and inner membrane. Outer membrane is a porous one, whereas the inner membrane is deeply folded. Now this inner membrane has a folds, and this folds increase the surface area of ATP generating chemical reactions. So why mitochondria has folds? Because it will increase the surface area for ATP generating molecules. Now what is the ATP? The full form of ATP is adenosine triphosphate, and that is an energy currency of the cell. Why it is known as energy currency? Because whenever the cell requires certain energy, it uses this molecule. So cell will get the energy. So that's why it is also known as energy currency of the cell, powerhouse of the cell, because mitochondria is producing, manufacturing ATP. Body uses energy stored in ATP for making new chemical compounds and for mechanical work. What all functions are performed by the cell? They all functions will be done with the help of energy. Now that energy is stored in ATP. They have their own DNA. So mitochondria has their own DNA and ribosomes, and that's why they are able to make their own protein. So mitochondria has ATP generating sites. They store this ATP molecules, and other than ATP, they have DNA and ribosomes, and because of that, they can make their own protein. We also refer mitochondria as powerhouse of the cell, and ATP is known as energy currency of the cell. Plastids are important organelles in plant cell. They are present only in the plant cell, and they also have DNA and ribosomes. So like mitochondria, they have their own DNA and ribosomes, so they can manufacture their own protein molecule. Apart from this, there are two types of plastids. One is chromoplast and other one are leucoplast. Chromoplasts are having a different color pigments. In that, chromoplasts are color pigment, whereas leucoplasts are white or colorless pigments. It has a pigment that is chlorophyll, and that is named as chloroplast. So chromoplast has a pigment chloroplast, and it is containing chlorophyll. So chlorophyll is important for the photosynthesis process in the plant. They also have orange and yellow color pigment. Now regarding chloroplast, internal organization of chloroplast has numerous membrane layers with stroma. They are similar to the mitochondria. So how the mitochondria structure is there, they are also having the same structure like mitochondria. So this is about chloroplast. Now leucoplasts are white or colorless. Basically they are primary organelles who will store starch, oils and protein molecules into them. So leucoplasts are colorless. Their function is to storage of all these materials, that is starch, oil and protein, whereas chromoplasts are colored pigment, and they have one pigment named chloroplast. So chloroplast has a chlorophyll into that is green color pigment, and it is responsible for the photosynthesis process in the plant. Other than this green color pigment, they do have orange and yellow color pigments. So plastids are only into the plant cell, and they have their own DNA and ribosomes into them. Vacuoles in the plant cell and animal cell has a special importance. In animal cell and plant cells, vacuoles are generally important for the storage of the material. They are storage sac, that means they are a bags type of structure, and they are used to store solid or liquid material. In animal cell, they are of small size, whereas in plant cell, they are having a large size. In plants, vacuoles are full of cell sac. The function of the vacuoles in the plant cell are they provide turgidity and rigidity to the cell. Second is important substance storage like amino acid, sugar and various organic acid and proteins. Storage will happen in vacuoles. Then in single cell organism and unicellular organisms, they are used to store some of the substance. In amoeba, they are used to store some of the food items, whereas in some other unicellular organisms, in vacuoles, they store some excess water and some waste material. Hope this video has solved your doubts, and in coming videos we are going to learn some more important concepts. So don't forget to subscribe, learn yourself. I'll see you in next video. Till then, keep learning, keep sharing.